Important events that occur in second week of development occurs at day 8, 9, 11, 12, and 13. On day 8, the layers of the blastocyst called trophoblastic layer and embryoblastic layer divides. The trophoblast divides into cytotrophoblast and syncytiotrophoblast, while the embryoblast divides into epiblast and hypoblastic layer. Along with this, two cavities are also formed, an amniotic cavity above the epiblast and a blastocele cavity below. Here is the diagrammatic representation of these changes. The cytotrophoblast appears to form as a mononucleated ring of cells with distinct cell boundaries. And above this cytotropoblastic layer forms syncytiotropoblast. This layer comprises of multinucleated cells with no distinct boundaries. The embryoblast also divides into epiblastic layer, which has elongated columnar cells, and a hypoblastic layer, which has cuboidal cells. A small amniotic cavity is formed above the epiblast, and a comparatively larger blastocele cavity is formed below the hypoblast. Day 9 is characterized by the formation of lacunar spaces and Hauser membrane, also known as exocoelomic membrane. Most of these changes occur at the embryonic pole and very few occur at the abembryonic pole. The lacuna appear as empty spaces in the syncytiotrophoblast, while the Hauser membrane originates from the hypoblastic layer and surround the inner lining of the blastocele cavity. Day 11 and 12 is characterized by the development of uteroplacental circulation and the development of extraembryonic mesoderm. We will also learn that the extraembryonic mesoderm divides into an outer somatic mesoderm and an inner splanchnic mesoderm. The uteroplacental circulation is developed when the capillaries inside the uterine wall thickens and dilate. These uterine capillaries are called sinusoids. Once these sinusoids are irritated by the syncytiotrophoblastic layer, they erode and the blood rushes inside the blastocyst to fill the lacunar spaces. The extraembryonic somatic mesoderm is originated by the inner layer of the cytotrophoblast, while the extraembryonic splanchnic mesoderm surrounds the embryoblastic cavities. Day 13 comprises of the formation of the primary villi, secondary yolk sac, and a chorionic cavity. The primary villi are formed when the outer edges of the cytotrophoblast invaginates inside the syncytiotrophoblast. Several connections develop between the somatic mesoderm and the splanchnic mesoderm, of which only one remain later, which is known as the connecting stalk. This connecting stalk later develops into the placenta. The blastocele cavity shortens and a new small cavity called secondary yolk sac is formed. The spaces between the mesoderms forms a cavity called chorionic cavity. This ends the second week of development. The blastocyst has now completely penetrated the uterine wall, while the uteroplacental circulation has also been developed. Let's have a quick recap of the second week of development. On day 8, the trophoblast divides into cytotrophoblast and syncytiotrophoblast, while the embryoblast divides into epiblast and hypoblast. Amniotic cavity develops above the epiblast, while the blastocele cavity develops below the hypoblast. Day 9 is characterized by the formation of lacunar spaces inside the syncytiotrophoblast and exocoelomic membrane called Hauser membrane, which is originated from the hypoblast. Day 11 and 12 is marked by the development of uteroplacental circulation and extraembryonic mesoderm. The extraembryonic mesoderm having two layers called somatic mesoderm and splanchnic mesoderm. Day 13 is characterized by the formation of primary villi, secondary yolk sac, and chorionic cavity.